Now we're going to move to chapter 4 and we're going to cover this topic of cells and organelles. So we're going to cover topics that we still don't have a lot of answers, like how the first cells appear. So we're going to cover some of those topics from a very general perspective. In addition, we're going to cover topics that you have heard so many times that are like organelles. So what is a mitochondria? What is a nucleus? And we're going to cover it from a very general perspective. In the following chapter, we're going to zoom in into each one of those organelles. So let's start. So we're going to talk about these fascinating debates like this one, which came first, the chicken or the egg. And when you talk about these debates at the cellular level, they become fascinating. So what do you think that it happened first in the cell? That you have the molecule of DNA, or what you have is the first proteins. Yeah. So in other words, what it came first at the cellular level, inheritance or function. And if you see, it's just too difficult to figure out. Yeah, because if you have DNA, the replication of DNA require proteins. Yeah. And if you want to have uh, similar proteins, you need to have inheritance of DNA. So not a simple question and not a simple answer. And in this class, what we're going to talk is maybe it could have been something different. Not DNA or proteins, but it could have been something that have both functions. And that possibly right now is not having, is not, is not currently doing that function. Yeah. So later I will explain that, but possibly RNA play a very important role. And RNA has both the a protein, a function, able to produce function, and is also able to produce inheritance. Yeah, so it's able to do both poorly, but it's able to do both. So we're going to talk about that, and we are also going to talk about the importance of lipids to create in the first cell. So we generally think about proteins or DNA, inheritance or function, but RNA is very important, and also the production of these different between what is inside. Yeah, let's put here in and outside. Yeah, that could have been also fundamental to create the first cells. And I'm sure that you read the textbook, or if not, I invite you to read it. And they start talking about this. Okay, so if you didn't read the textbook, or if you read it, try to think about this. In the first cells, try to organize from the first to the last these different components. Yeah, what happened first? DNA, RNA, protein, metabolism, cell membrane. What do you think that happened first and which one happened last? Okay, so take a few minutes. I already told you some of the potential ideas that we have right now, but try to organize it and try to think why should have been one first than the other one. So here we have a timeline of the events related to the history of the planet. And why this is important, because to understand what happened in the first cell, yeah, how the first cells emerge, we need to understand that they emerge in a context. So we know that in the past, something that it was key is the formation of the earth. And here in the beginning, the earth was very unstable. All the volcanoes, we need to create uh, an environment that it was stable to be able to support life. So when that happened, we think that that happened around 4.2 billion years ago. Yeah, that we start having um, the life cycle, the water cycle that we more or less know right now. We have less volcanic activity, and the conditions become more favorable. Then we think that at that time, between 4.2 to 4.0, the prebiotic chemistry appears. So we have the first macromolecules forming. And then when we have all this, and when we think that something called the pre-RNA war started. And this RNA, as I said before, has the ability to both have inherent potential as well as functions. Then it creates this RNA war. So maybe the first living organisms have this potential to produce this metabolism, yeah, based on RNA. And then we got the life as we know it right now, that is more specialized. The inheritance had DNA that is more stable than the RNA, and the proteins, yeah, they were able to do more functions, just specializing in both. So we have, as we will study in genetics and also in this course, what we see is that we find a lot of relics of RNA everywhere, and we find so many types of RNA. And in molecules that are very important, we find pieces of relics that tell us that maybe RNA was very important in the beginning, although it's still very important, but it was important generating the metabolism in the beginning, the inheritance, and also many functions. Okay, so this happened 3.6 million years ago, and then after that we have the diversification of life. Well, we have all the life as we know each other, that multicellularity appear, and we have all the different uh, life forms as we know each other. Okay, so if you want to know more about this, we have here a link that they talk about all this information. Okay, now let's move to one of my favorite talks in biology that I will show it to you in a second. Okay, so here, if you one day find that you're in the mood to listen in more detail how life appear and all the knowledge that we have about it, here is one of the worldwide experts that is talking about that, and is telling you all the relics why we think that the war started with RNA, and the importance of lipid membranes to create these first cells. Okay, so this go beyond this course, so and I get too excited watching this video, so I'm not going to talk a lot about that because this go beyond the course objectives. But if you're interested about that, I leave you the link so you can watch it. So here you can see the organization of this lecture that is based on what you find in your textbook. And in the beginning, we're going to talk about how the first cell come to be. We're going to later talk about some properties and strategies of the cell. Then we're going to briefly, very briefly, I'm not going to talk about the difference about bacteria, archaea, and eukarya in a lot of detail because you have heard a lot about that. I'm going to skip some sections and tell you a little bit of the story of some specific that you might not know. If you want to remember this, if you don't remember this difference, the textbook um, have that explained in a lot of detail. Then we will talk about the internal membranes um, and this topic, and believe it or not, will be at least one third of the semester, talking about the different organelles that have membranes that are very important and how membranes are very important for the traffic at the cellular level. Then we will talk about several limitations on cell size that we start in the previous class. Now we're going to talk I'm going to show you our examples, and then I will talk about the structure of function of cells. Again, I'm going to skip some sections that you have here a lot in previous courses, and we're going to focus on new things. So let's start to this origin of the first cell. So these are some diagrams that I wanted to show you that appear in your textbook. And here, currently, we think about cells like membrane plus DNA. Yeah. However, what appear first? Yeah, or they appear independently, membranes and these molecules for inheritance. And in this course, yeah, we are going to talk about diagrams like this that you have seen all your life. But I also want that you think about this. Maybe cells look like cities, yeah, that you have cars going in one direction or one's in another direction and things moving. Cells, as we are going to show you using live imaging, yeah, so now we track what is happening inside of each cell, and they're very dynamic. They're vesicles moving from one region to another one. So it's not only what happened if it was the DNA or a protein or the membrane, but how that metabolism, how that dynamic appear is fascinating, and we still have many questions. And I just wanted to tell you that it's not a simple question, and now I'm going to show you how your textbook tried to simplify this important question that we still don't know all the answers. To simplify this question that we still don't know completely, your textbook divided in 4 different stages that this is what I'm going to explain. So the first primitive cells, in the beginning, they have simple organic molecules, and your textbook show it like this. Then we we have the polymerization of these simple organic molecules. So here you can see that. And in the third one, the emergence of macromolecules like DNA and RNA. That this is what your textbook is showing here. And finally, the encapsulation of the first living cell. That this is what you can see here. So let's talk a little bit of each one very briefly in the following slides. So I'm going to start talking about the first stage. In the first stage, here in your textbook, they show you these different geometrical shapes that what they are represented is different inorganic compounds. And these inorganic compounds, they start forming more complex molecules, yeah. And those complex molecules, they start forming organic polymers that it become that are fundamental for life in our planet. And I'm not going to explain this in a lot of detail because you studied that in high school, and also you studied that in introductory biology, and these are the experiment of Stanley Miller. So if you haven't studied this, if you don't remember this, you can go to your textbook and read it in more detail. I'm going directly and going to ask you a question to know if you remember that experiment. Okay, so let's read it together. Experiment by Stanley Miller in 1950 demonstrated that the first organic molecules on Earth, and they give you these different options. You can stop the video and read it and try to answer it. It's not very complicated. Try to think about the answer and try to see if you remember what is this experiment. So let me show you the answer. Here you can see the answer is A. So if you remember, the experiments of Stanley Miller, they demonstrated that the first organic molecule on the Earth were composed or appear thanks to the early atmosphere that contained hydrogen, methane, ammonia, and water vapor. So if you see hydrogen, it's very simple molecule, methane, CH4, very simple, ammonia, and water vapor. Let me see if there's a space here for me. Okay, yeah, and so all of them very simple molecules that they were able to produce more complex molecule like DNA, like polymers of like amino acids, and these kind of molecules. So if you see, all the other questions are wrong. Why? Because, for example, B, the simple molecules were composed of simple amino acids. No, actually, car amino acid is an acid, yeah, that has an amino group. So look, here you can the hydrogen, the methane, the ammonia, all these water vapor are the main constituents, main inorganic molecule that allow to produce amino acids. So it's actually in the other direction. These are too complex. RNA and nucleic and DNA are more complex molecule. Here, if you read this one, they talk about extraterrestrial environment, and more than saying yes or no, this is not about the experiment of Stanley Miller. Don't talk about this. And here is a combination. If you read E, this is a combination of both D and E. Yeah, so this is doubly that has two mistakes, not only one. So the best answer is A. I hope that it was clear. Now I'm going to explain very generally, very briefly, yeah, what is the Stanley Miller experiment, and I hope that that if you want to give a look, you can read it in more detail in the textbook. Okay, so just to briefly talk about this, I want that you observe these diagrams that you might know, and you can see the different components. So here is one component of this experiment of Stanley Miller, that is the electrical discharge. Here you find another environment where you have how they simulate the atmosphere here, and here you have some of those chemicals. Here there's in a different in temperature, and here is where you trap some of the new chemicals that are formed. Yeah, and here is where they accumulate. So what they did is with this experiment, they replicate the possible environment for the synthesis of organic compounds. So here they took an electrical discharge, that is the one that produced the energy to break the chemical bonds and form new ones. And here they took, here are the different molecules that were very simple, like water, ammonia, hydrogen, hydrogen 2. And what it happened is that the electrical charge and all these different simple or compounds are going to mix and form new molecules. And what they found is that when they try here, what happened, yeah, is that they found simple organic compounds. Yeah, for example, they found glycine and alanine, that I'm sure that you remember them, that there are two of the simplest amino acids. And this is just one example. They found that if you modified this the environment simulations, you can produce different compounds. So this favored the theory that life, at least this step, is not as difficult as we thought. Yeah, just this simple experiment can show that with these basic molecules, yeah, you can produce much more complex molecules, and so the first stage is easy to achieve, the transition from the first one to the second one. So now that we know how this happened, we can move into the next step, that is the taking these organic monomers, each one of these subunits could have unite to another one and form these polymers. Okay, and I'm going to show you an example, the example of your textbook, that is the DNA and RNA. So if you remember monomer and polymers, yeah, mono, one, poly, many. So for example, if you have adenine here, yeah, that is very important for the formation of these nucleic bases. So what you're doing is, if you have one, you can have multiple of those one, and you can attach them as Lego parts, and you can start getting more complex and more complex components. So you form something simple like a Lego part, and then you assemble it into something more complex like a spaceship, yeah, from basic units. So this is how we hypothesize that more complex macromolecules like DNA start forming. So now that we have macromolecules, at this point, we say like these macromolecules like DNA and RNA are able to store information. And to me, this means that this is how life starts. However, here the question is, but how you're able to copy, to replicate the DNA. Yeah, and if you remember genetics, okay, the DNA need to replicate, but to do that you need enzymes and you need so many proteins. So you need proteins that copy the DNA, Yeah, proteins that open the chain, or other ones that release the pressure so the DNA doesn't break. And again, something very weird. Some of them need to put RNA molecules there so the polymerase work there. So it doesn't make sense to do that step there, but it's there. Why we need to replicate DNA to have RNA? And of course, you can guess that this is a hint of an important process. This is one possibly of the relics of this world based on RNA. Okay, so we're going to talk about that. So definitely right now, when you see all the biological organisms in the planet, most of them will work like this. There is central dogma where you have DNA that is in charge of the perfect storage of information, very stable. Then you have a transitory stage where you store information, but it's more unstable. That is the RNA, and finally at the end you have the function. So the idea is that although this is how we find it now, maybe in the past we use something different. Yeah, so maybe, as I said before, RNA seems to exist before. And one of the evidence is that not only DNA but also RNA are both macromolecules that store information. Yeah, so not only DNA store information, here you can see that RNA store information. In addition to that, to produce the process of uh, protein synthesis, you need to all go to the RNA. Yeah, so RNA seems to be fundamental for both. It's able to do the storage process as well as the protein synthesis. Okay, and in the following slides, I'm going to show you that there are some molecules of RNA that are able to do also functions. Yeah, so in other words, RNA is a not such a good form to store information. Yeah, DNA is much better, and it's a not so good way to have functions. Yeah, but it's able to do both. So maybe life start with not such a good and specialized molecules, but the one that is able to do both at the same time. Okay, so I will explain that in 1 second, and I will show you a video. But now I'm going to ask you one question about the RNA world. Okay, so let's go into that. Okay, so the question, if you read the textbook, you already know the answer. It's also very easy. So what is the RNA world hypothesis? Okay, so you can stop the video and try to guess which one is the correct answer. And in a second, I'm going to show you a video. I'm going to play a video that talk about this RNA world hypothesis.
Base pairing also gives RNA chains a second special ability. When placed in water cool enough for base pairing, but without enough free nucleotides for replication, chains will fold up and base pair with themselves. The end result is a complex shape with certain sticky bases pointing outward because they weren't able to find partners. These sticky, outward-facing bases can cause unique chemical reactions by interacting with other molecules in their environment. A folded chain of RNA capable of guiding a specific chemical reaction is what we call a ribozyme. Some ribozymes break certain molecules apart, others join certain molecules together. A ribozyme's specific function is determined by its specific shape, and its shape is determined by its sequence. If a mutation changes a ribozyme's sequence, the shape can be modified, and so can its function. When ribozymes were first discovered, scientists wondered how difficult it would be for random chains of RNA to evolve legitimate survival functions. Imagine, for example, a ribozyme that could build nucleotides out of molecules it finds in its environment. Across multiple generations, natural selection could promote and refine this ribozyme because the chain would tend to have access to more free nucleotides than its rivals, allowing it to replicate more often. To explore this idea, researchers at Simon Fraser University produced a large group of random RNA chains and examined them to see if any happened to be able to make nucleotides. Surprisingly, some actually could, but they weren't very efficient. Researchers selected out the successful chains and then used a lab technique called PCR to quickly replicate them with slight random mutations. After just 10 rounds of PCR, followed by selection, highly efficient nucleotide building ribozymes evolved. These are molecules with the lifelike ability to actively participate in their own survival. These ribozymes, and many others produced through similar experiments, are beginning to blur the line between living things and simple chemistry.
Okay, I hope that you enjoyed that video and you can watch it all. There is more information there, but I think with that information, with the clip that we watch, is enough to answer this question. So, what is the RNA war hypothesis? So, in all this class, I have been telling you that is the idea that not DNA but RNA, yeah, were the first self-replicating molecule. Yeah, so I will show in future slides, it seems that the DNA is just a better way to copy the genetic information. And for that reason, it played a key role in the origin of life. So we will see in future years, there is a lot of evidence that the RNA war could have existed, yeah, but we will see in 20 years if new evidence appear that make it strong this hypothesis or that a new one appear. Okay, so now let's go and see more information from your textbook. Okay, so to summarize things, I just told you that RNA may have been, so it's a hypothesis, be the first informational molecule. And just to summarize things, to show you what they talk in their textbook, I told you one important piece of information. Yeah, so, and you can read this here, and if you want, you can write it down. And it sounds very complicated, but I'm going to try to explain it. So I'm going to explain what is this deoxyribonucleic acids and ribonucleotides, okay? So if you remember chemistry or remember your introduction to biology, the difference between RNA and DNA, so the D and the R. So the R come from a type of sugar that is ribonucleotides. So you said ribose. Here you see a small difference in the sugar, yeah, in the molecular formula, while the other sugar is deoxyribose. So you see here the difference, yeah. So why I'm showing you this? Because what the textbook is trying to show you is like to produce DNA, yeah, here the deoxyribo, you need to have RNA components plus specific enzymes. So, so although we go in this direction, yeah, for the central dogma, to produce the materials for DNA, the pathways, yeah, it seems to be relics, seems to be very old, and the pathways to produce DNA, the materials require the production of first RNA, and from those components you produce DNA. Okay, so that is fascinating. And it seemed to be showing that, do you see here the RNA word, that maybe there was no need of DNA in the beginning, that the RNA was copying itself as we watch in the video. The second part of this evidence, of this diagram, so here we said that the information storage, you didn't have DNA, you have RNA, and for the function here you see that instead of proteins, maybe you have used also RNA. So here you can also type or writing down this, and especially this ribozymes. Okay, so if you took genetics, there are many type of RNAs, and this is one that is very interesting because it's able to catalyze, yeah, to perform certain enzymatic reactions. In other words, it's able to be able to storage information, but also to produce some functions. And one example of that is the formation of peptide bonds that occur during translation. So it seems that RNA could have done both. Yeah, so I show you that before of the mother war of the central dogma that we have, there is evidence indicating that RNA could have appeared, could have done the functions of both DNA and proteins, and DNA and proteins later appear, and they were just more efficient on both information storage, as shown here, as well as be able to perform different functions. And when I hear about this, I said, this doesn't make any sense. Yeah, how RNA is able to do both, and how something, how RNA is able to do something like that, but if you do it inefficiently, how life could have appeared. Yeah, and I want that you think in a different way. I want that you think that in evolution and in life, we generally think that everything starts perfect, but it could have been started slowly, not as good, and start improving and improving and is still improving. Okay, and this could have happened both with the inheritance and with the function. And I'm going to use a metaphor that is not perfect, but for me was useful, and is the use of computers. And uh, I don't know how many of those of these storage devices did you know. I don't know if you, when you're watching the video, you know that there was CDs. We don't use them anymore. Some old computers still have them. I have my PhD thesis in still in some CDs. Uh, Maybe you have this one, and you have heard about this one, right? Yeah, it has now here. Right now we have the cloud, OneDrive. There are so many things, yeah. And maybe you have seen in museums. The first thing that I used was this, one of these discs, and before this, there were cards that have holes. Yeah, they were not perfect, yeah, but they were useful, and they break a lot. They didn't have a lot of capacity, but we went to the moon, and I still, it's incredible. We did a lot of things before all these things appear. Yeah. So maybe life didn't have to have the perfect molecule that it was the DNA, or let's say perfect is a very reliable molecule that it still work after millions of years of evolution. Most of the organisms use DNA. And in the same way, I wonder you think about functions. So yeah, so uh, if you have used, I don't know how many type of word you have used, yeah. With Word, we use the functions, we write text, we write essays, scientific papers, yeah, all the function is there. Uh, Maybe, I don't know, when are you watching this video? I don't know if this is not anymore, we don't use it anymore. I don't know if you were able to see this or this. I think everything for me start with one of these ones, and it was really bad. It was really bad. I finished many documents and they didn't save. It was very unreliable, but it was better than this. Yeah. Yeah. I don't know. My first in elementary school, I think I had one of these ones. Yeah. Because you were the common way to do it. And every time that you made a mistake, there was no way to delete it. You need to use like a special pen to delete things. And if you made too many mistakes, it was like a paint. You have to repeat all the work. Yeah. So the ability to so the that right now when you made a mistake that we have with Word and delete, backspace, we don't have it there. So there was, although the first one that I used has a lot of problems, it was still much better than the word processors of the past. The you know the So the main point here, and I don't want to make this long, I think you got the idea, is that life didn't need to start perfect, and there was a gradual improvement in the same way that there is a gradual improvement with computers. Yeah, it's this metaphor is not perfect, but I wonder you think about that, both the storage of information of computers and the way that they produce functions like using applications, they have been improving and they will continue improving. Okay, and the same thing happened with life. Okay, I hope that you enjoyed this part of the class. Now we're going to move into the next section, that is the liposomes.